All right. All right. Now I'm ready. So we begin part 2. This is terminology that I labeled way back when as possibly helpful. It is actually helpful. I wasn't lying to you. I just wasn't giving you the whole truth now. Aldose sugars, aldose carbohydrates are reducing sugars. They are reducing carbohydrates. And ketose sugars, ketose carbohydrates are non-reducing carbohydrates. That becomes important when you're dealing with paper 3 stuff, and it may or may not be important for paper 1. For paper 2, it comes up far less often. But aldose and ketose sugars, the distinction between them is, stay out of the shot, that in an aldose sugar, the compound has a carbon-oxygen double bond that is located at the end of the carbon chain as an aldehyde group. A ketose sugar has the carbon-oxygen double bond located in the interior of the chain, a ketone group. So, an aldehyde group, carbon double bonded to oxygen with a hydrogen here, and the rest of the chain. A ketose sugar, the end is whatever, and then whatever, and the carbon double bonded to oxygen, and it continues. Whatever else these carbons are attached to doesn't matter. If the carbon double bonded to the oxygen is in the interior of the chain, and its neighbors are both carbons, that is a ketone. If the carbon has only one carbon neighbor, and the other neighbor is a hydrogen or an oxygen, it's not a carbon. That is an aldose sugar or a reducing sugar. It's an aldehyde group if it is carbon double bond oxygen with a hydrogen. It's carboxylic acid group when it's carbon double bond oxygen and a hydroxyl group, OH. More about reducing sugars in a moment. So, glycosidic bonds. The name glycosidic bonds or glycosidic linkage or glycogen bonds refers to the bonds between carbohydrate monomers. It is formed by a condensation reaction. The hydrogen interacts with the hydroxyl group on the other side, forming water, and you end up with a 1,4 glycosidic linkage. If we're talking about glucose to glucose, and you get maltose. Alpha glucose to alpha glucose, and you get maltose. So that is a glycosidic linkage, glycosidic bond, glycogen bond. It is still a condensation reaction. You circle the hydrogen and the hydroxyl group. The oxygen that is closer, the hydroxyl group that is closer to the oxygen as part of the ring, keeps the oxygen. The oxygen with less interesting stuff, the carbon with less interesting stuff around it, loses that hydroxyl group. And that's where you form the bond. Simple carbohydrates are when you put together a monosaccharide and get a disaccharide. Those are simple. Complex carbohydrates are polysaccharides. The more units you add, the more complicated it becomes. The more parts you have, the more complex the machine. So a simple carbohydrate has very few parts. A complex carbohydrate has a lot more parts. So, that is just the food definition for mono, di, and polysaccharides. Simple, complex. When you add monosaccharides and to disaccharides, this is your chart. Glucose plus glucose gets you maltose if you are adding 2 alphas. If you are adding 2 betas, you get cellobiose. If you are adding glucose and fructose, you get sucrose. And you need to know the bond configuration as well for the purposes of the Cambridge exam. They sometimes ask about the bond configuration. They ask about the numbers themselves. Usually on the paper 1 more so than the paper 2. But they still ask about the bonds. And because they ask about the bonds, you must know them. So, glucose and glucose connect by a 1,4 glycosidic linkage. If it is an alpha 1,4, then it is 2 alpha glucose molecules combining, where they are in the same orientation. The hydroxyl groups are both down or both up on either side. In cellobiose, beta glucose, they're in a trans configuration. The hydroxyl group is on the opposite side. The hydroxyl group is up, and the hydroxyl group on 4 is up, and the hydroxyl group on 1 is down, or vice versa. But for your purposes, alpha is always down. Alpha is always down. Alpha is always labeled in the down configuration. That way, the 6 carbon is always pointing up. Just for the sake of drawing it, to make it easy, alpha is always down and the 6 carbon is always up. That is always the case. The 4 carbon is the one where the hydroxyl group can switch, where it could be up or down. Sucrose is an alpha 1,2. Glucose is in the alpha 1. The alpha down. 1 down. 2 beta. It's the alpha 1,2. Alpha 1,2 beta. The fructose is in a beta position, which means they flip. They are at a 180 degree from each other. That means something because they are in a certain configuration of shape. When things are a different shape, enzymes cannot act on them. Enzymes only work on certain configurations of shape. So when the shape changes, when you flip a molecule upside down, that shape is different enough that the enzyme doesn't recognize it and the enzyme doesn't work. So glucose, maltose, which is glucose and glucose, cannot be broken down, can be broken down differently from sucrose, which is glucose and sucrose, which is a flipped fructose. That flipping means it is a different shape. Different shape means the same enzyme will not work. Enzymes must work on the same shape. That will come up a lot more. Glucose and galactose forms lactose. That always forms in the beta 1,4 configuration because of the way that the the hydroxyl group is shaped and flipped in the Fisher projection model. It always works that way. So glucose and galactose can only come together in the beta configuration, which means it's treated by the body the same way that cellobiose is. We cannot break it down. It is like fiber to us. Unless you have lactase, which is an enzyme that specifically breaks down glucose and galactose. This must be cut by lactase. If you lack lactase, If you do not have lactase persistence throughout your life, you are what's known as lactose intolerant. You cannot process and digest lactose. And it acts as fiber, which acts as a salt. And what does water do? Water follows salt into your gut tube. Water follows it into your gut tube in large quantities, and it comes out the other end in large quantities. That is why people with lactose intolerance have gas, bloating, and bad diarrhea when they drink milk. Lactulose is a combination of fructose and galactose. We cannot process this at all. There is no enzyme for this. This is a medication used in the treatment of hepatic encephalopathy. It's used in the treatment of liver disease, brain swelling due to liver disease, because it draws fluid and stuff out of the liver. It draws fluid and stuff out of the body and basically takes away the toxins that would normally be processed by the liver. And it prevents the liver from having that damage done to it further. It also pulls out ammonium just straight up. That's just one of its properties. So that's something you don't need to know for the Cambridge exam. I'm probably going on too much. No, you don't need to know that for the Cambridge exam. Ignore that. In fact, we should probably just cut that out from the video. But lactulose is a medication used to cause massive diarrhea for beneficial effects. No one likes being on lactulose because it is very, very runny, very, very uncontrollable. Diarrhea is horrifying. So, we don't need that. We do need this. This is alpha versus beta glucose. I'm sorry, it's the 1 carbon. It's the 1 carbon that flips, not the 4 carbon. My apologies, my apologies. It's the 1 carbon that flips, not the 4 carbon. Still full of lies. So the 1 carbon flips. That's the one that flips. The 4 carbon is always that configuration. My apologies. The 1 carbon flips. So if the 1 hydroxyl group is down, the 4 hydroxyl group is down always, because it's always down from the 6, and the 1 is down from the 6 as well. These two are in the cis configuration. That is alpha. If the beta configuration, the hydroxyl group is up compared to the 4 down. The 1 is up, the 4 is down. They are in the trans configuration, across from each other. This is the flat model. This is the chair projection. The reason this matters is because when you try and connect these two to each other, you try and connect one of these to one of these, you end up connecting this to here. That works just fine. Connecting that hydroxyl group to that hydroxyl group works just fine. Here, in order to connect that hydroxyl group to that hydroxyl group, you must flip it upside down. You must flip the whole molecule upside down compared to this one. So you flip one upside down and you can connect them. So you have to flip them 180 after another. So the beta glucose has a 180-degree flip every monomer. It goes up, flip, 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 as you add monomers to the chain. That is beta. For alpha, you can just keep adding 1, 2, 3, and it will just spiral. It will make a spiral pattern. This is the alpha versus the beta chains. This is the beta chain. This is why it stays straight. Because these hydroxyl groups are flipped over one on top of the other, the oxygen hates the other oxygen. And when you combine two strands like this, they will zip right together. they will have polar interactions with another molecule, one right next to another. So they will zip together into these little fibers. Versus the amylose, which is the alpha 1,4 glycosidic bond. All of these hydroxyl groups are on the same side. They will start to pull toward each other through hydrogen bonding, and they will curve. And they will curve into a spiral. In order to remain in a configuration that is stable, they will curl into a spiral, and you get curly pasta or whatever that stuff is called. I don't even rigatoni, whatever. I think that's this. Never, not a. The structure of cellulose itself is massively important for this, and this is tested anytime they talk about plant cell walls. The structure of cellulose is directly tested when they talk about plant cell walls. What about their hydrogen bonding allows them to be water porous, allows them to be fibrous? This is how it works. This gets you points on the Cambridge exam. They form together in these long, straight beta chains. And the hydrogen bonding between each chain of the hydroxyl group on the 6 carbon. I'm sorry, the hydroxyl group on the 2 carbon. The 2 and the 6. Allow for the hydrogen bonding between the individual strands. These individual strands zip together like a Ziploc bag. But there is room here because water is a small molecule and these are polar bonds for water to just zip right through. So this is porous. This is porous because it is forming individual strands that may cross over each other to form a meshwork that forms the cell wall. And water can just go right through the meshwork. And water can also but water cannot interact directly with the individual fibers themselves. The individual fibers themselves are watertight, but the space between the fibers are not. So cell walls are porous, but they are not at all water repellent because they are made of hydrogen bonds. They are made of polar interactions. So they are and do form adhesive, they do adhese to water. They have an adhesive property. Water can adhere to the cellulose structure, allowing them to attach and adhere to the walls of xylem during transpiration. That is how it works. That is how you get those points. Because the cellulose itself has hydrogen bonding, because it is made of polar molecules, water can adhere to cellulose. Because it is made of fibrous strands that individually are watertight, but as a group form strands and meshworks that are full of pores. It is not watertight. Water is freely permeable across cell walls. So this is a property of cell walls in plant cells, directly related to the structure of beta glucose chains. That is how it connects together. That is how Unit 1 connects to Unit 4. That is how you get maximum points from a question on Unit 4. So they have great cohesive strength together because of all these hydrogen bonds. Hydrogen bonds, when you add them all up, are very strong. Individually, a hydrogen bond is very weak, but when you add a lot together, they are very strong. Oh no, stop that. Okay. Polysaccharide energy storage. This is important for paper 3 and a lot of paper 1 stuff, so we're going to go over it a little bit. It's slightly less important for paper 2. Starch is the plant energy storage molecule for carbohydrates. It is the carbohydrate storage for plants. Amylose is a single straight chain with no branches. It spirals because it is simply alpha, 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 alpha monomers. Just a straight chain of alpha monomers of glucose. And because of that hydroxyl group, on the same side every time, it bends into a spiral. This is the starchy starch. This is the fluffy stuff. Amylopectin is amylose with chains that are branched at the 6 carbon. It has a firm texture. It holds together well. This is the firm starch. So white potatoes, red potatoes, stewing potatoes. Nutritionally though, starch is starch. It's all carbohydrate. It'll all make you fat. Because carbohydrate is the easy source of energy. You break that down into glucose. When you have too much glucose, you break down the energy, you use the energy to build fat stores. Glycogen is the animal version of starch. It is not starch. Glycogen is not starch. Glycogen is the animal storage molecule. Structurally, it is more similar to amylopectin, but it has more chains closer together. But it's the same thing. It is 1-6 chains, 1-6 branching chains. No branches, few branches, more branches. The chains are alpha 1-6 linkages. You need a special kind of enzyme to break this linkage because it's not the same shape as a 1-4 link. It feels different. It's a different shape, therefore it would fill a different active site on a protein. So when I told you that carbohydrates are C1H2O1 ratio, there's actually 2 lies embedded in this. That is an individual carbohydrate monomer. The individual carbohydrate monomer is typically 1:2:1 ratio. But when you take a carbohydrate monomer and a carbohydrate monomer and make a carbohydrate dimer, a monosaccharide, and make a disaccharide, what do you take out? You take out water. You take out H2O. So, when you have a disaccharide, the ratio is C1H2O1 minus H2O. Whatever that ratio is, times whatever number, minus 1 H2O. It always comes out to that because you took out the water. You took out 1 H2O in order to combine 2 monomers. Now, there's another thing that you can do to this, and you still call it a carbohydrate. You dope it with nitrogen. And when you dope it with nitrogen, you get chitin. Chitin is N-acetylglucosamine, which makes the shells and the shell of crustaceans and insects. Beetles, wings, crabs, etc. That is the polymer of N-acetylglucosamine. That is chitin. There is also chitin found in mushrooms too. That's part of the cell wall structure there. But you don't really need to know too much about that. That's for me. There you go. Let's talk about lipids. Fatty acids is the simplest form of lipid. And these are amphipathic lipids. Amphipathic is as definitions go. Definitions for amphipathic. It has a polar head and a non-polar tail. It has a polar and non-polar end. There are two ends to this molecule. It can exist in polar and non-polar conditions. Fatty acids have the formula of carbon, hydrogen 2, and oxygen 2. They always have two oxygens. These are just fatty acids. The reason they only ever have two oxygens, no matter how many carbons and hydrogens they have, is because they end as a carboxylic acid. C double bond O, bonded to OH. Carboxylic acid with an R chain, a carbon chain of variable length. It doesn't matter how long it is, it doesn't matter how many double bonds it has, it doesn't matter anything else. That is your fatty acid. And there are many different types of fatty acid, many different names for them. You don't need to know that. You just need to know a fatty acid is a carboxylic acid with an R chain of a carbohydrate. A carbohydrate chain. Carbon and hydrogen, and that's it. Saturated versus unsaturated. A saturated carbohydrate has all the hydrogen it can handle. It cannot have any more hydrogen. It is all single bonds. It has only single bonds connecting it to the next carbon in the chain, and therefore can hold two hydrogens on every carbon, except the last one, which gets 3 because it's special. So every carbon gets at least 2. In an unsaturated double bond, in an unsaturated fatty acid, there is a double bond. One double bond makes it unsaturated. Polyunsaturated makes it more than one double bond, multiple double bonds. So, the saturation is referring to, is it full of hydrogen or not? So that is fatty versus not. The properties of this are related to van der Waals forces. Van der Waals forces are not the same as hydrogen bonding, but you can think of it in the same idea as hydrogen bonding. Hydrogen bonding is the interaction of hydrogen and usually oxygen. It's polar to nonpolar, hydrogen to a polar group, electropositive to electronegative. Van der Waals forces are the interaction between just nonpolar neutral molecules. It is a weak interaction. The more kinks in the chain, the more bending, the more interaction that two things can have. They can clamp down or loop over and link with other things just by geometric shape, overlap and pull. And so unsaturated, sorry, my apologies. That's only when we're talking about polypeptides. I'm confused. Van der Waals forces are different, my apologies. Unsaturated fatty acids, the kinks in the chain force them away from each other. And when the nonpolar groups are interacting with other nonpolar groups, they tend to clump together. So a saturated fatty acid chains, the longer chains will group together in long straight chains that make very efficient packing. And when they pack efficiently, they become solids at low temperatures. Or they become solids at lower temperatures. So you get solid fats like meat fat and butter fat and egg fat, saturated fats like this. Unsaturated fats are your oils. When you have kinks in the chain, they don't group together as well. They are inefficiently packed. A solid object is very efficiently packed. And inefficient packing allows more movement and more wiggle room. More wiggle room is the difference between a solid and a liquid in terms of phases of matter in general. So unsaturated chains means they force each other away, and there is more distance between them and more wiggle room, which makes them liquid at lower temperatures. So oils and vegetable fats and vegetable oils tend to have more unsaturated and polyunsaturated fats. Which one's better for you? Well, unsaturated fats are better for you. But if you're talking about engineered polyunsaturated fats like olestra, which was a thing back in the 80s, that was an engineered polyunsaturated fat that caused horrible diarrhea, greasy, fatty stools in people who ate it. Olestra chips, fat-free potato chips. What a wonderful concept, until you started pooping very large amounts of greasy, fatty stools because you couldn't process